transformation was first done by a guy named Griffith. He injected mice with strain 1 of bacteria. The mice died. He then injected mice with strain 2 of bacteria. The mice lived. Next he injected mice with he killed strain 1. Again the mice lived. Lastly he injected strain 2 and he killed strain 1 into the mice, but they died. This determined that some part of a he killed strain 1 must have incorporated into strain 2. Important words to this chapter are nucleotide, double helix, and DNA. A nucleotide is a monomer of a nucleic acid polymer, while a double helix is a structure of DNA is a twisted ladder shape. Lastly, DNA is inheritable genetic information of an organism. Back to the experiments. Avery repeated Griffith's steps, but he was more clever. He included protein-destroying enzymes in one trial and DNA-destroying enzymes in another. This concluded that protein could not be the genetic material because transformation still occurred. More important words are the bases. Thymine is a pyrimidine that bonds with adenine. Adenine is a purine that bonds with thymine. Guanine is a purine that bonds with cytosine. Cytosine is a pyrimidine that bonds with guanine. In case you were wondering, a pyrimidine is a single-ring nitrogenous base, while a purine is a double-ring nitrogenous base. Finally, to Hershey and Chase, they wanted to determine if protein or DNA was the genetic material. They made a batch of a virus with radioactive sulfur. Next, they made a batch of a virus with radioactive phosphorus. They let each batch infect non-radioactive cells. They removed the shells outside of the cell by using a blender. This measured the radioactive of a cells. At the end, only the cells with radioactive phosphorus had radioactivity inside. This concluded that DNA was the genetic material. Next we have DNA replication. This is the process of copying DNA. The first step is helicase separates the strands. Next, DNA polymerase will pair free nucleotides with their complementary bases. This is done with hydrogen bonds. DNA polymerase then will attach neighboring nucleotides to each other with covalent bonds. Lastly, DNA polymerase will proofread and fix the mistakes. Lastly, the steps of translation. Translation is when RNA is made into protein. Translation begins with the attachment of a ribosome and the first tRNA to a start codon. Then the ribosome moves along the mRNA transcript. The polypeptide elongates as a amino acid is added for each codon. Finally, the ribosome arrives at the stop codon and the completed polypeptide is released.